Good afternoon, everyone. For me, it's a pleasure to be here showing some of the results of our last project, which is a culture-independent approach to understand the microcystis colony composition and diversity across the time. First, we need to know what is a microcystis colony. Microcystis is one of the most studied cyanobacteria associated with toxicity. This cyanobacterium is unicellular. They can make aggregates. During the aggregation, they evolve to multicellular structures called colonies. Additional to the cells of microcystis, we can find in the colonies a complex structure of associated bacteria. That was the things that which Eve was mentioned before. So in this study, we just focus in the microcystis colony. As we can see here in this picture, we can see the cells of microcystis that are coming from a culture, but also we can see associated bacteria that belong to the Caulobacteriales. So essentially, using colonies of microcystis that we can recover from the Lake Champlain and also from Brazil, and using genomics, we want to understand the diversity of microcystic genotypes occurring in the lake. We also want to know if specific genotypes of microcystis are related to a particular associated bacteria, and if those associations between microcystis and the associated bacteria are stable across the time. With this in mind, we sampled this sampling in Lake Champlain during the last two summers, 2017 and 2018. We went to the lake and we took the water with a plankton net with the help of Natalie and Nico and also Alberto. We concentrate the colonies of microcystis and we isolate in the lab approximately 200 colonies. When we had these colonies, which are between 1 and 3 millimeter size, we did for each single colony DNA extractions, getting really small quantities of DNA, but enough to do the genome sequence. Here, I can show you the number of sampling points that we did across the last two summers, 2017 and 2018. Every bar represents a sampling time point. And the numbers represent the number of colonies that we sequence. We, in total, sequence 82 microcystis colonies. We also are doing the same with samples from Brazil with the help of our collaborator, Alessandra Guiani. And we actually had some colonies ready to do genome sequence too. The results that I'm going to show today are related with the samples of the colonies that were isolated just in Lake Champlain. So one of the main findings when we start to work with microcystis colonies, when we did the DNA extractions and start the genome analysis, what we found is that microcystis colonies are associated with multiple bacterial genotypes. This is a cladogram where when we classify the sequence of microcystis and of the colony based on the sequence composition and the coverage. In this part, we had a nice cluster of sequence that belong to microcystis. So, but additionally, we had also sequence that belong to other taxa in the colony, like Phenylobacteriums and associated genomes that doesn't have a taxonomical classification. But we can talk in details about these taxonomical classifications later if you are interested. So in this part of the project, we are just going to focus in those sequences of microcystis related with microcystis that are in the colony. So when we just talk the sequence of microcystis and you forgot about the sequence of the associated bacteria, we start to doing similarity analysis in these 82 microcystis genomes. Using this similarity analysis and population structure analysis, we were able to classify these 82 genomes in 8 genotypes. In this picture, what we have what we had is a similarity matrix where the colors are proportional to the similarity between microcystis. As we can see, we had 8 genotypes just in the Leyland plains, and 2 of them have been reported before, which is important because show how a culture-independent approach allowed to recover a higher diversity of microcystis genotypes in a lake. So we asked about the toxicity of these microcystis that are in the lake. Most of them had the genes related with the biosynthesis of microcystin or NCY operon, excluding one that is related with microcystis flossacui. So this is, I think, really sad because most of the genotypes that I identified here had the genes, but doesn't mean that they are producing microcystin. Other thing that we can see is when we are studying the genotypes of microcystis is that how many of them are in each sampling point, are they concurrent? In this chart, what you had is the time points, and each color represents a microcystis genotype. Basically, what we had is that several microcystis genotypes are concurring across time in summer 2017 and 2018. We had some of these genotypes, like the G7, that is present in all the sampling points of 2017 and some of the 2018. So we recovered, as I say now, 6 genotypes in 2017 and 8 in 2018, 2 new ones in 2018, which is important. So now, once I just start to work with the associated genomes and just forgetting then about the sequence related with microcystis, what we found is that here again we had the associated genomes per each colony of microcystis. We take all the 82 colonies and we did a super assembly or co-assembly of this sequence, and we got that there in the samples, in the colonies, there is around 70 different genomes of associated bacteria, complete and incomplete genomes. So what we did was estimate the relative abundance of these associated genomes, excluding microcystis per each colony. Using this matrix of relative abundance, we did a distance-based redundancy analysis, including abiotic factors like pH and temperature, and biotic factors like microcystis genotype. So what we found is that abiotic factors like pH and temperature doesn't have significant effect in the composition of the associated bacteria in the colony, but biotic factors like the microcystin genotype have an important effect. When we plot the relative abundance or the results of the distance-based analysis, we had here different points. The distance are based in the relative abundance of the associated bacteria, excluding microcystis, and each point is a colony. What we can see here is that the colonies are clustering based in the genotype. That means that colonies with the same genotype had a similar community of associated bacteria. So that is important because maybe it's talking about the long association between these associated bacteria in the microcystis genotypes. So as a take-home message, I just want to say that I was quite surprised because Leyland Plain hosts a diverse community of microcystis. So we had 8 genotypes, just doing colony sequence. Before using just cultures of microcystis, we only had 2, and we had 6 new ones. Other thing that we can see is that microcystis colonies are associated with multiple bacterial genotypes. Additionally, the composition of associated bacteria communities depends on microcystis genotype. I would like to say thank you to Shapiro Lab members, Alessandra Gianni, that is the person who collaborated with us in Brazil, Natalie 14, Alberto, and Miriam that are helping us with the sampling, and also Elisa Tocheva, that is the person that is working with the microscopy of the Microcystis cultures.
Thank you. Any questions for Olga?
Congratulations for your presentation. My question is, do you think there is a kind of a symbiotic relationship between the bacteria and the cyanobacteria? And if it is yes, what could be the relationship? I mean, probably some kind of commensalism or some other relationship?
I think the answer at the first question is yes. And what we know based in just in genomic data is that there is some genes related with biosynthesis of vitamins, like thiamines, and the other things that we can see is that there is, for example, sphingommonas that is related with the degradation of microcystins that probably is helping to the maintaining the equilibrium in the colony of the microcystis cells. So this is probably other thing that we found working with it is that there is some kind of associated bacteria that are being co-evolved with the microcystis genotype, which means that probably they have been there since a long time, and they are giving some kind of help to microcystis.
Okay, one more question, if possible. Do you think that in some special condition you can delete all the associated bacteria in order that you have only cyanobacteria cells?
Well, people have been working with us in the past, and they are using many multiple doses of antibiotics. So they can at least try to recover microcystis at zenith, but it's almost impossible, like maintain there without the associated bacteria in a long period of time. They probably, as a consequence of the treatment with the antibiotics, they are going to die. So it's not that well. I think it's almost impossible to remove the associated bacteria without affecting to microcystis.
Thank you.
Well, thank you.